Hello, everyone. Can everyone hear me? Can you put on the chat if you can hear me, please? Can you put a yes on the chat if you can hear me? Thank you. So welcome to our session on A-level biology, so year 13 respiration. And we'll be covering quite a lot of topics today, which can be quite complex. So we'll try to take it slowly and try and explain as much as possible. And you can stop me, you can tell me to rewind any parts that you don't know at any point, and we can go over that again. So I'll have some scheduled breaks in where I'll ask you guys if you want me to recap everything that we've just gone through to make sure that you're still understanding, if that's okay. So our timeline will look something like this, but we don't necessarily have to stick to it. So first of all, we'll be looking at respiration overall. So we'll be looking at things like the importance of respiration, what is respiration, and where it takes place. Then we'll have a mini break. Then we'll go on to look at some glycolysis, and then this might not take as much long, and then we might have some lunch and then go on to the rest of the parts of the respiration cycle. That's okay with everyone. So that's just a brief summary of what we're going to today, but we might not necessarily stick to this timetable depending on how fast or how slowly we go through it. So to introduce myself, I'm Nimisha. I'm a third-year medical student at the University of Sheffield. I'm currently intercalating to do a BSc, so I'm taking a year out of medicine to do a different degree, and now I've signed up to do some teaching as well. So that's fun, and that's all there is about me, really. So how does this work? So we'll be giving you top tips throughout this presentation. So we'll be looking at what the exam wants you to answer when they ask you certain questions. We'll have some practice questions along the way. We'll also have some exam practices, so looking some past paper questions towards the end. And then we also have Ask Ace, which is another tutor who's on this chat. So if you have any questions at any point that I can't get to through my presentation, you can ask on the chat and she'll answer for you. And then this will also be recorded, this presentation, so you can watch it back at any point, so that you can always have it in your revision as well. Okay. So the topics we'll be covering are the importance of respiration, the sites where respiration takes place, the events of respiration, so the 4 steps of respiration, the importance of the coenzymes that are involved. So we'll talk a bit more about what these are in a bit. And then we'll also look at the chemiosmosis theory, which is very important in creating ATP and generating ATP to release energy. And factors that affect respiration rate will not necessarily be covered in this presentation, but this will be in your practicals and things. So you'll have covered that in your course anyway. So that's great. So before we start, can I ask you to vote if you've already done a respiration before, or if this is completely new to you, just to see what level everyone's at, and then we can start going through it slowly. That's okay. So just put A if you've already done it, and B if this is the first time. That's great. So I can see some of you have already done it and some of you this is completely new. So we'll look through it as if we're completely starting this new, and we can go through it quite slowly and try and understand everything as we're going along. And if you would like me to recap as we go along, please let me know and we can do that as well. Okay, so to start off with, I'm just going to ask you a quick question. Why do we need a respiration in our body? If you could put your answers on the chat, please. No one else can read what you message, so it's fine. Yeah, so someone's put to produce ATP, that's correct. And we produce ATP, and that's just one form of energy that we're producing, because what we need as living organisms is to produce energy. Sorry. So all living organisms are made of cells. So every single part of our body is made of a single cell, and each cell requires energy, and which is what someone has replied that we produced in the form of ATP. So that's just the form that we produce, release our energy in. So sorry for saying produce, but energy is always released, not produced. So can anyone give me examples of when we need energy in our bodies? Yeah, so someone said for active transport, that's correct. We do need ATP for active transport. Anything else? Yeah, so any kind of anabolic, so someone's written cell division, so that's correct, because any anabolic reaction, so that's any reaction which requires you to break down something, you require energy for it. And someone else has written regulatory forest. That's excellent. So we need energy when we're trying to maintain homeostasis. So when we're trying to maintain our body temperature and regulate our hormones and things like that, we need energy. And we also need energy to make important molecules. These are all great answers, guys. So as we have here, so we need it for our very own basic movement. So even to blink or to talk as I am now, we need energy. So any kind of movement requires energy. Sorry, I'm just trying to get my pen out. So any kind of energy, any kind of movement requires energy. And then, as someone said before, regulatory processes such as maintaining body temperature or just general homeostasis requires energy. And then anabolic reactions. So example would have been, as you said before, cell division, that also requires energy. And then transporting substances across membranes. So as someone said, active transport is also a form that requires energy to cross the membranes. There are also other types of transport that require energy, but not necessarily in the form of ATP, but we don't need to know it at this level. Cool. That were all great answers, guys. Thank you for contributing. So I'll just move on to the next slide. So before we talk about what and where the respiration takes place, can someone tell me what exactly is respiration? Yeah, so someone's written it's the release of energy from glucose. That's correct. So what I'm trying to tell you is that respiration is not ventilation. So what's a common misconception is that respiration is the same as breathing in and out. That's not correct, because respiration is what takes place in our cells, not necessarily what happens when we're breathing in and out, and that would be ventilation. So in an exam question, they might try and trick you and ask about respiration, but you might think they're talking about ventilation, so try to not get them confused. So it's not breathing in and out. So it actually occurs in all our living cells. And as someone said, it releases energy from glucose. So it doesn't necessarily have to be glucose. It can be breakdown of organic molecules. And this is then when we convert the chemical energy, so it can be in carbs, which would produce glucose, and proteins, which would release amino acids. And sorry, just getting my pen up again. So proteins can release amino acids, which can then go on to generate energy. And then we can also have lipids. Lipids go on to produce, also get involved in the Krebs cycle, but this is a cycle that's only used when our main source of energy, which is carbohydrates, as someone said, this releases glucose, and that's our main source of energy, and that's the most important one. So what do we turn all this potential energy to in our body? Someone's already answered this before, but I just wanted to reiterate it. What form do we generate energy in our body? Yeah, ATP. So we generate ATP. Sorry, it's not clicking. Yeah. So ATP, or known as adenosine triphosphate, is a molecule which has 3 phosphates attached to it, and that's why it's called triphosphate. So say if I drew the main part as a pentagon, and then the 3 phosphate molecules are these 3 circles. So it's the addition or displacement of these. So this bond here between the second and the third phosphate molecule, this is actually quite weak, and that's when we can generate or break down ATP to release energy. So this here is when we have, say, just this part would be ADP, which is our diphosphate, which means it has 2 phosphates attached. And the whole thing together is ATP when we have our energy molecule. So what happens is when ADP joins onto this phosphate molecule that's just hanging around in our cell, this is when we generate ATP. And it's the process of respiration that we can generate this ATP in, and we'll look into it a bit more detail in a bit. So the next thing I wanted to ask you guys is the equation of respiration. Does anyone know the general equation of respiration? So we can break that down into more steps. So what are the 2 substrates that we need in respiration? So someone already said one of them, so I'll write that down. So we have glucose as one of our substrates, and yeah, as someone has correctly said, oxygen is the other one. Sorry about my handwriting. The screen's not helping me. And then what are our products when glucose and oxygen combine? So carbon dioxide, and then water and energy. So this energy doesn't necessarily have to be in the form of ATP, but so in this case, we'll just call it energy for now. So this is just releasing of energy because this is a catabolic reaction, but we won't necessarily call it ATP because it doesn't always have to be ATP in this reaction. So just moving on to the next part. This is the equation written out much more nicely than I have with my handwriting. And this is the chemical formula for it. So this is a balanced equation which shows that glucose, which is a 6-carbon molecule, so I'll just write that down. It's a 6-carbon molecule, and this will come, this will be important later on when we looked at the individual steps of respiration. And then this combines with our oxygen, which then forms our carbon dioxide, water, and the energy. And we'll see how these products are formed during the steps of respiration as well. So we'll see where carbon dioxide comes from and where this water comes from as well, and where these get used up as well. Okay. And this again is just the equation written down much nicely than I have. Okay. So moving on, where does respiration take place? Yeah, so as most of you have said, respiration takes place in the mitochondria of a cell. Yeah, but we'll find out a bit later that one step of the respiration actually takes place outside, but we'll go on to it in a bit more detail. But first of all, we're going to look at the structure of a mitochondria. And so the mitochondria are rod-shaped. So as you can see, it's shaped like a rod in this image, with a diameter of 0.5 microns to 1 micron. So this is not meant to say meters, it's micrometers. So it should say micrometers or microns. So make sure you write either one of microns or micrometers in exams to clearly show you know what you're talking about. So they might ask you what is the structure of a mitochondria in the exam. So you say it's got a diameter of 0.5 micrometers to 1.0 micrometers. And then, sorry, it's not clicking. So we've already said this multiple times, so I'm just going to skip through it. The role of the mitochondria is to generate ATP. And this is the main part where we generate ATP and not necessarily energy as a random term. So this is specifically where we form ATP, which we need for our human reactions. Oh, sorry, that's a sneak preview. So I've got a labeled mitochondria here. So if we can have a guess at what each part of them is. So can anyone guess what number 1 is, although you might have had a sneak preview? So I've labeled the different parts of the mitochondria, and they may ask you to list them in the exam. So yeah, the first part is our outer membrane, which is on the, obviously as the outside of the mitochondria, our organelle. Our next bit we have is the intermembranous space. So that's between the outer membrane and the, what's number 3? If the other part's the outer membrane, what's the other part called? Yeah, the inner membrane. So we have the inner membrane and the outer membrane, and in between it, we have the intermembrane space. And this becomes important in the very last step of respiration, which is oxidative phosphorylation. And we'll go on to it a bit more detail in a bit. And can anyone tell me what number 4 is, what these folds in the inner membrane is called? This part here. So that part, yeah, is called the cristae. And the cristae is very important, and I'll explain it more in our next slide. And then number 5 here is pointing at the blue solution. does anyone know what the solution in the middle of the mitochondria is called? So it's called the matrix, and this is also involved in respiration, obviously, but we'll also talk about the function of it on our next slide. I just wanted to label all of them first so that we have an idea of what they are before we look at why they work. And then 6 here is pointing at a ribosome, which is involved in protein synthesis. And then does anyone know what number 7 is? These swirls of. So these swirls are actually DNA. So mitochondria has its own DNA because it can form its own proteins. And this is called mitochondrial DNA. And you might hear of people with mitochondrial disease, and that's due to a problem in their DNA in the mitochondria. So I'll just clear all my drawings and go on to the next slide where we can talk a bit more about what the function of each thing is. So, as we said before, as a normal cell is, our mitochondria itself is made up of 2 phospholipid membranes. So the outer membrane is, sorry, I'll just get my pen up again. So the outer membrane is here, and as you can see, it's on the outside, and our inner membrane is here. And that's what's highly folded to form these cristae. So, does anyone know which part of is it the inner membrane or the outer membrane that is smooth and permeable to tiny compounds? Yeah, it's the outer membrane that is permeable to smooth and tiny compounds because it's on the outside. So we want stuff to come in and out of the mitochondria, but we only want the stuff we need to come in and out. So we don't want large molecules that might be harmful to the mitochondria to come out. So that's why the outer membrane acts as a guard of some sort. So it's like a gating where you can decide what it wants to come in and what goes out. So that leaves the next part, which is which part of the membrane is rolled up to form the cristae and is less permeable. Yeah, the inner membrane. So the inner membrane is important because this is where the electron transport chain starts. And does anyone know what the enzyme that forms ATP is called? So the enzyme that forms ATP is called ATP synthase, which is quite simple because if you think about it, it's just synthesizing ATP. So synthase just means to synthesize, and we're synthesizing ATP. And it's on our inner membrane that we'll find these enzymes, which we can pretend this is an extension of my membrane over here. And this is where we'll find these ATP synthase molecules that generate our ATP. And so it's very important in respiration because this is where most of our ATP is formed, which we will use for all our processes such as movement and maintaining homeostasis and things like that. And then nextly, which part of the mitochondria has a low pH due to a lot of protons? Just take a guess. There's no right or wrong answer. I mean, there is, but I don't think I would have known either. So do people think it's the matrix or the intermembrane space? Yeah, so someone said the intermembrane space. So the intermembrane space is where we have a lot of protons, and we'll find out why it has a lot of protons in a bit, but this is just giving you a rough idea of what's involved. So our intermembranous space here, which is the bit between the outer membrane and the inner membrane. So don't forget, intermembrane space means between outer and inner membrane. And this has a low pH because protons, or H plus ions, which has lost an electron, so now it's just a singular proton, it increases the acidity as there's a lot more positives. And so this becomes, the pH decreases when something becomes more acidic, and so you get a low pH. And then finally, we have our aqueous solution within the inner membranes that contain the enzymes, ribosomes, and circular DNA. Does anyone know what that is called? Yeah, the matrix. So the matrix is just the name given to this aqueous solution. So the solution right in the middle of our mitochondria. So this blue liquid here, that contains all our things we need for generating ATP. So the ribosome, which creates our proteins, such as our ATP synthase, which we'll need for ATP generation, and DNA, which we need to transcribe onto the mRNA by the ribosome. So they're all involved in this protein synthesis process. That's why the matrix is important, to keep it all in there. So in the exam, they might not necessarily ask you to label, say, which part of a mitochondria is this, but they'll want to know more things like, why is something like this? So for example, the structure of the mitochondria is very suitable to its function. So can anyone tell me why we have the cristae on the inner membrane? So the inner membrane is highly folded, as you can see here. So I'll draw an extra diagram. So the inner membrane's like this, whereas the outer membrane's like this. So this is the inner membrane. Can anyone tell me why something might be highly folded to form these cristae? And cristae is just a name given to the folds. Yeah. So you said it increases surface area, and that's very important, because if something we can increase surface area, and as I said before, we have our ATP synthase molecules, which sits on these membranes. So if we can pack a lot more of them into the same space, this means that we can generate a lot more ATP than we can generate into the same space if, say, it was on the outer membrane, which is just straight. So we can see in the same space I have here, I can only put 3 ATP in the same space I've managed to put on 5 in the inner membrane. So that's why having the cristae, which increases the inner surface area, is a lot more important. So that's just summarizing what I've just said. So the cristae, which is the membrane folds on the inner membrane, it allows the membranes to store a lot more synthase enzymes. And so that's good, because we need a lot more, we can generate a lot more ATP. So the amount of mitochondria in a cell, so not just the mitochondria itself, but the amount of them in a cell can also vary depending on what type of cell it is. So can someone give me an example of a cell which requires a lot of energy? Yeah, so muscle cells, which we obviously need for movement, are a lot more active than, say, fat cells, which are just there to store our energy sources. So our muscle cells will have a lot more mitochondria than other cells do. And I'll clear all my drawings again. Is everyone still following? Shall I do a mini poll? Can you put A if you're still following and B if you're not. That's great. Everyone's still following. That's great. Okay, so we'll move on to the next part, which is a couple of questions. So my first question is, what number replaces the x in this equation? So I'll run a poll, so you don't have to put your answers in the chat. So just put it A, B, or C, D corresponding to the answers on the slide. So when we balance this equation, which is the glucose plus oxygen makes carbon dioxide plus water plus energy equation, what replaces the x in this equation? That's great. So everyone's put 6, which is the right answer. So I'll just explain it in case anyone's not quite understood why. So we have 6 here, which is the correct answer. So the reason why this x and this x and this x becomes a 6 is because this is a 6 carbon molecule. So when we balance the equation, the x needs to be 6 in here, so that it's 6 carbon molecules, because there's no other carbons in this. There's no other carbons in this equation. So this definitely needs to be 6. So in this case, we end up with 12 oxygens. But as you can see here, we end up with 12 oxygens, but we'll wait for that in a second. As you can see here, we have 12 hydrogens on the glucose molecule, but on this side, we currently only have 2. And so to balance the equation, we do what, 6 times 2, because there's 2 sets of hydrogen for each one. So 6 times 2 would be 12. So we've now made our carbon molecules are both 6 on each side. Yeah, carbon molecules are 6 on both sides. We have our hydrogen, which are 12 on both sides. And then we have our oxygen molecules. So on this side at the moment, we have 8 oxygens because we have 6 here and 2 here. But on this side, we have 12 oxygens and 6 oxygens, which makes 18. Sorry, I think I've just confused myself. So we'll just quickly go back to the equation from before. Sorry. Okay, so in our equation here, we have our 6 oxygen molecules and our 12. Yeah, sorry, that makes sense. I've just confused myself. I'll go back. Sorry for confusing everyone. I'll just start from the start again and explain it a bit better this time. So to start off with, we wanted to balance the carbon molecules. So I'll draw a little table here. So this is the left side and this is the right side. We had 6 carbons on this side and we need to make the right side 6. So we make it a 6 here. So that gives us 6 carbons on both sides. Then we want to balance the hydrogen. So we have 12 on this side, put a 12 here. And we want to balance that. So what times 2 gives us 12? It would be 6. So 6 times 2 gives us 12. And now we need to balance the oxygens. So on this side, we can see 6 times 2, it gives us 12. And then 6 times 1 gives us 6. So altogether on this side, we have 18 oxygens. But we already have 6 oxygens here. So we can take that off and that leaves 12 left over that we need to fit into this oxygen molecules. And so 6 times 2 would be 12. And now we have balanced both sides of our equation. And that's why we put a 6 in front of each of these numbers. I hope that's made sense and I haven't confused you even more. Okay, now I'll move on to our next question. It's a little less complex. Which of the following is not found within the matrix? The DNA, a ribosome, a lysosome, or an enzyme? And I'll restart my poll. So just give me a second. That's great. Everyone's gone for the right answer. It is indeed lysosome. So just to go over the answers, lysosomes are organelles which are involved in cleaning up our dead cells. So it breaks down our organelles and is involved in basically clearing them up, and is also involved in phagocytosis, which is, our body's immune response. But then we definitely have DNA in our mitochondria, which is our mitochondrial DNA. And then we definitely have ribosomes, because I said it's involved in protein synthesis, so things such as ATP synthase. And then we definitely have enzymes, because ATP synthase is the enzyme which generates ATP, which is found inside the matrix. Whereas lysosomes are found in the cytoplasm of our cell. So it's not found within the matrix. Okay. Let's clear all my drawings again, and we'll move on. So now we're going to move on to the individual stages of aerobic respiration, but we'll have a mini break before that, so you can have a breather and ask any questions if you want to as well. So I'll meet you guys back at 1:45, if that's okay. And ask any questions on the chat if you want to as well. Actually, I'm going to extend that break just because I feel like 5 minutes isn't long enough. So we'll restart at 10 to, if that's okay. Hello everyone, hope everyone's back. Should I run a quick poll to make sure everyone's still awake? So you can vote for whichever one you would like. I just want to make sure everyone's still awake. Shall we? Great, everyone seems to be back, so we'll continue as we have before. And if you have any questions throughout, make sure to ask Ace and they'll answer for you as well. Okay, so I noticed a question before that I didn't get a chance to answer, and it was if photosynthesis is the opposite of respiration. And you can think of it like that because the products are the opposite, because in respiration, we get glucose plus oxygen makes carbon dioxide plus water, whereas in photosynthesis, the process is in the opposite direction, but the actual steps involved in photosynthesis are quite different. And you'll learn that when you go on to photosynthesis, if you haven't already learned it. So the actual physical steps are quite different, but the overall equation is the direct opposite of the other one. So I hope that answered your question. So we'll continue with the stages of aerobic respiration. So the primary substrate of our respiration is glucose, as we've mentioned a couple of times now, and that comes from the carbohydrates. And aerobic respiration breaks down glucose with the help of oxygen. And on the other hand, anaerobic respiration, I'll just write it here. Anaerobic respiration is without oxygen. Has no O2. So there's no O2 involved. So we're going to look at the four main stages of aerobic respiration in the rest of our lecture today. So does anyone know what the first step of respiration is? Yeah, it's glycolysis. So glycolysis can occur in both aerobic respiration and anaerobic. And this is because it takes place outside the mitochondria, and so you don't need oxygen for it. And we'll go on to where it takes place in a bit. So that's another question. So what's the next part of respiration after glycolysis? Yeah, it's the link reaction. And the link reaction is only in aerobic respiration. So there is no anaerobic respiration here. So this one is only aerobic. And what's the next step in our aerobic respiration? Krebs cycle, yeah. So next up is Krebs cycle. And this is also, like link reaction, is only aerobic. And then what's our final step of respiration? Yeah, it's a big word, but it's oxidative phosphorylation. And this is the last step of only aerobic respiration. Anaerobic respiration works slightly differently, and we'll cover that at the end after we've gone through all of the steps of aerobic respiration. So glycolysis happens in both aerobic and anaerobic respiration. And whereas link reaction, Krebs cycle, and oxidative phosphorylation is only aerobic. So I want to make sure you're clear on that. Cool. Going on to the next slide. Clear my drawings first. Okay. So now that we know what the steps of respiration are, we should know where it takes place in a cell. Does anyone know where glycolysis takes place? Yes, it takes place in the cytoplasm. So this actually doesn't take place in the mitochondria. So this is not in mitochondria. And that's important because that's the reason why it can be both involved in aerobic and anaerobic respiration, because it doesn't need oxygen and it can take place in the cytoplasm without the mitochondria. So what our next step was link reaction. Does anyone know which part of the mitochondria that the link reaction takes place in? Yeah, it takes place in the matrix, which was the aqueous solution in the middle of our mitochondria, which contained our DNA, our enzymes, and our ribosomes. And next up, we have Krebs cycle. Does anyone know where that is? Yeah, this one's also completed in the mitochondrial matrix. And then finally, we have our oxidative phosphorylation. Does anyone know where that is? Where that happens? Yeah, so it happens on the inner membrane of our mitochondria, and the cristae actually refers to the highly folded membrane itself. So where it takes place is the inner membrane, and cristae is referring to how it's highly folded. So it takes place in the mitochondrial inner membrane. So as you can see, the top one, which is glycolysis, takes place outside the mitochondria, and these are all inside the mitochondria. So somewhere, at some point, the molecule from glycolysis needs to enter the mitochondria, and we'll look into it in a little bit more detail. So let's clear all the drawings again and go on to the next slide. So to summarize glycolysis, this is our first stage of respiration, which occurs in both aerobic and anaerobic. And this is when we convert a 6-carbon molecule called glucose. So we saw before the equation for glucose was C6H12O6. So as you can see here, it's a 6-carbon molecule. And then we split that into 2 pyruvate molecules. So pyruvate, as you can see, is a 3-carbon molecule. So you can see the 3 carbons here. So we split that into 2 3-carbon molecules. We get a net gain of 2 ATP, and I'll explain why it's a net gain in a bit. So we get a net gain of 2 ATP at the end, and we get 2 reduced NAD. I'll explain what is a reduced NAD in a bit as well. So these are our products of our glycolysis. So 2 pyruvate molecules, net gain of 2 ATP and 2 reduced NAD. And those are our 3 products, 3 different products that we get from glycolysis. So now we'll look at the individual steps of glycolysis. Okay. So the first step of glycolysis is phosphorylation. So glucose is phosphorylated by 2 ATP. So, and this forms a fructose bisphosphate. So I'll annotate as we go along. So as we know, an ATP molecule, as I drew before quite badly, is say this is our adenosine bit, and then this is attached to our 3 phosphate molecules because it's triphosphate, which means 3 phosphate groups. And what happens is this bond between the 2nd and 3rd phosphate breaks, and we donate this phosphate molecule we have here to the glucose. And because there's 2 glucose involved, 2 ATP molecules involved, this happens twice. So they release 2 phosphate molecules. And this then attaches onto our 6-carbon glucose molecule. So as you can see here, this is an inorganic phosphate, and this is an inorganic phosphate. And that forms our fructose bisphosphate. And bis just means it has 2 phosphate groups attached. Okay, so I'll just clear all that again and summarize. So we get our 2 ATP molecules coming in. And 2 ATP both release one for inorganic phosphate group each, which makes it 2 ADP because they both lost their third phosphate group. So this gains one phosphate group on this side and another phosphate group on this side to create our fructose bisphosphate. And that's the first step of glycolysis, which is known as phosphorylation. It's called phosphorylation because we are giving our phosphate groups from the ATP molecule into the glucose molecule. So we're phosphorylating the glucose to make it a fructose bisphosphate. So this is a 6-carbon molecule with 2 phosphate groups. Yeah. I hope that's okay with everyone. And then clear all my drinks and go on to the next slide. Then what happens is lysis. So this molecule we have here, the fructose bisphosphate, is actually a highly unstable molecule because it has the phosphate groups attached to it. And so when ATP is broken down, it releases energy, right? Because that's what we use ATP for. Breaking down of ATP, breaking that bond releases energy, in which case we need that energy which makes this molecule very highly active, and it's quite unstable. So it breaks down into 2 groups. It breaks down into 2 groups known as triose phosphate. And all that means is that it's a 3-carbon molecule with a phosphate group attached, because triose means 3. So it's a 3-carbon molecule with a phosphate attached. And the reason why it breaks down is because it's a high energy molecule, because of the energy released from the ATP when we phosphorylated it. So it's a high energy molecule, it's unstable, and so it immediately breaks down into 2 3-carbon molecules known as triose phosphate. And this is the second step of glycolysis, known as lysis. Is that okay? Let me know if I'm going too fast, then I'll slow down as well. And then in our next step of glycolysis, so this is just summarizing what we said. So fructose bisphosphate, which is our high energy molecule, which released the energy from the ATP when it was phosphorylated. It breaks down into 2 3-carbon molecules known as 2 triose phosphates. So, and then oxidation occurs of this triose phosphate, and it's transferred, and the hydrogen is removed and transferred to this coenzyme called NAD. And we'll look into what exactly is a coenzyme later, because NAD is quite significantly involved in the whole process of respiration. And this gives us 2 reduced NAD. So this is the balanced equation for that. So 2 NAD makes 2 NADH. And then this is just balancing out the equation. So we'll look at what that means all together in a bit. So that's our third step, which is oxidation. So just to annotate what's happening. So we have our 3-carbon molecule, which is our triose phosphate. Our triose phosphate is oxidized, which means it has a carbon removed from it. But there's these coenzymes called NAD, and coenzymes are enzymes which don't necessarily get involved in the reaction itself, but are assisting in the reaction. So this releases, this gains a hydrogen from this triose phosphate, and it becomes reduced. So gaining a hydrogen means that it's reduced. Gaining a hydrogen means that it's reduced. It's a reduction reaction. So this is also known as reduced NAD. Okay. Whereas what's happening to our triose phosphate here is that it's actually oxidizing because it's losing a hydrogen. So gaining a hydrogen is known as reduction, and losing a hydrogen is known as oxidation. So our triose phosphate molecule, which is our 3-carbon molecule, is oxidized. And our NAD, which is a coenzyme, gains that hydrogen and it becomes reduced NAD. And this actually happens twice. So that's why we get 2-NADH as our end product. And then within the same step as well, what happens is the phosphate from the intermediate substrate molecules is transferred into the reaction and it forms the ATP. So, and it goes on to be used in the next stage of respiration. So I'll actually draw out the whole step here to make it a bit more understandable. So does anyone remember what the first step of glycolysis was called? Yeah, so our first step was phosphorylation. And this was when we had our original glucose molecule, which we know is 6 carbons. So let's write 6 carbons. And then we get 2 ATP molecules coming along and donating their inorganic phosphate molecule. So we get 2 ADP molecules. And then this produces our fructose bisphosphate, which means it has 2 phosphate groups. So I'll just draw out what that means. So this is our original glucose molecule, say. And then what happens with our fructose bisphosphate is that we add on 2 phosphate groups. So let's say the star is the phosphate group. So we add on these phosphate groups. But because there's a release of energy when we break down this ATP, this fructose bisphate is very unstable. It has a lot of energy and it's very unstable. So it wants to break down into triose phosphate. So 2 lots of triose phosphate. Which is a 3-carbon molecule. And then what happens here actually happens twice over. And that's why we get a net gain of ADP and things. So just to explain it. So our end molecule in our glycolysis is pyruvate, as you remember me mentioning. So pyruvate is our end molecule of glycolysis, and this is what then goes on to the link reaction. So how this triose phosphate goes to pyruvate is by oxidation. So as I mentioned before, the NAD comes in and then gains a hydrogen and then becomes NADH. And then in the same process, we have dephosphorylation, which is when 2 ADP molecules, which were released previously, and then come on, and then they want to gain the inorganic phosphate group. So this here, I'll draw up my little molecule again. So this is our triose phosphate with the phosphate group still attached, which only has 1 phosphate now because it's split in the middle before, and now it has 1 each. And so the ADP molecules come in to gain that phosphate group, and they produce ATP. And so they produce 2 ATP molecules. So as you can see here, in this reaction, we produced 1 pyruvate molecule, 1 NAD molecule, NADH molecule, and two ATP molecules. But what actually happens is, because this is only one triose phosphate we've done this reaction for, but we need to remember there's reactions happening twice over because there's two of these 3 carbon molecules now. So we need to double everything. So we have 2 pyruvate molecules, 2 NADH molecules, and 4 ATP molecules at the end. But then we actually say it's a net gain of 2 ATP because we've lost 2 ATP here. So we lost 2 here and gained 4 here. So we say it's a net gain of 2 ATP. Is everyone still following? Would be a good question at this point. So can you put A if you're following and B if you would like me to go through that whole step again? Okay, so everyone's still following. That's great. Don't hesitate to ask me if you want me to slow down and go over it again. I don't mind at all. So just to summarize, our first step in glycolysis is phosphorylation, which is when ATP releases its phosphate group to produce the fructose bisphosphate. And then this undergoes lysis. So I'll write lysis here. Lysis when it breaks down into 2 molecules. And then oxidation occurs here as the NAD, which is the coenzyme, gains the hydrogen. And then dephosphorylation occurs here as we lose, as we get rid of the phosphate group. So at the end, we're left with a 3 carbon molecule without the phosphate group attached. And this is what then goes on to the link reaction. So as you can see, it's phosphorylation, then lysis, then oxidation, and then dephosphorylation. I hope that's all made sense. And we can go over it again at the end if anyone wants me to. Okay. So sorry, I'll clear all my drawings to start off with. Okay, so I'll run a poll again, just to see if everyone's still awake. What is the net gain of ATP during the process of glycolysis? So it's asking for the net gain, not the total amount of ATP gained. Yeah, everyone's gone for the right answer. So I'll close the poll. So yeah, the answer is indeed 2, because as we learned, we lose 2 ATP at the start in phosphorylation. And then we gain 4 ATP during dephosphorylation because the whole process is happening twice over. Phosphorylation, and that's why we get a net gain of +2. So net gain of +2. Okay, so I'll clear that and then go on to the next question. Sorry, technical difficulties. Okay. What is the order of the steps in glycolysis? No, sorry, I should be running the poll. There we go. Yeah, everyone's gone straight for the right answer. So that is correct. We have phosphorylation, lysis, oxidation, and then dephosphorylation. And then just as a quick question, can anyone tell me what are our products from glycolysis? And how many of them do we have of each? Yeah, we have 2 pyruvate molecules. What else do we get as our end products? Yeah, we also get 2 NADH and 2 ATP, and those are our 3 end molecules. And these are all important in the next steps of respiration. So we'll move on. So ATP is used to make the glucose more reactive, and that's what we saw happen as we saw the fructose bisphosphate was a highly reactive molecule. Sorry, that does not look like it says fructose bisphosphate. Fructose bisphosphate is a highly reactive molecule and it's very unstable, and that's why it quickly lysed to form the 2-triose phosphate. And this was when we used 2 ATP to produce the 2 fructose bisphosphate so that it would donate its inorganic phosphate molecule. Organic phosphate molecule. So as you might have seen me noting it down, inorganic phosphate molecule can also be written as PI. And I would recommend writing out the full thing in your exams, but if it's easier for you to note it down when you're revising and things like that, you can also note it down as PI, which just means inorganic phosphate. And then we got 4 ATP, which are produced right at the end in our dephosphorylation process. And that's how we ended up with our net gain of 2 ATP. Okay. Clear all my drawings again. So our end product of the glycolysis is pyruvate. And this goes on to the next step of our reaction called the link reaction. And this can either, so our pyruvate molecule can then either go on to do anaerobic respiration or aerobic respiration. So if there's oxygen, this can go on to do aerobic respiration in the mitochondria. So if there is oxygen is present, then it can go into the mitochondria and conduct aerobic respiration. But if there's no oxygen, then it does anaerobic respiration within the cytoplasm. So it doesn't go into the mitochondria. In the cytoplasm. Okay, so pyruvate is rich in chemical energy and then it contributes to our next step, which, can anyone remember what the next step after glycolysis is called in respiration? Yeah, so the next step is the link reaction. So as we'll remember, the link reaction actually happens inside the mitochondria. So we need to somehow get the pyruvate into the mitochondria to conduct this link reaction. And this actually happens by active transport. So let me just write that down. So it needs this transport protein and a small amount of ATP. And we know any transport that requires a small amount of ATP is known as active transport, because as we remember, our double membrane is only permeable to tiny compounds. and it's smooth and it's only permeable to tiny compounds. And pyruvate's reasonably big because it's a 3-carbon molecule. And so we need a transport protein and small amount of ATP, so via active transport, to get into the mitochondrial matrix, as it needs to cross both the double membrane. So once it's inside the matrix, then it can carry out the link reaction. Okay. I'm just going to quickly run a poll, and can you put A if you have any questions so far, if you have any questions, and B if you have no questions and you're happy to go on. Okay, we have a question, so do you mind sharing it on the chat to me? Yeah, to put it on the chat to everyone, and we'll address it. Just want to make sure everyone's on board before we go on to link reaction. See if you want to put the question to me on chat. Ah, okay, it was a mistake. That's fine. That's all right then. If everyone's okay still, then we'll go on to the next part, which is the link reaction. But if you do have any questions, just put it on the chat to ask Ace or me, and we'll get it answered. Okay, so moving on, we'll go on to talk about the link reaction. So sorry, I keep leaving my drawings on. So links, it's called the link reaction because it links the glycolysis and the next step, which is the Krebs cycle. And the steps are summarized here, and we'll break it down and draw a diagram because I really like diagrams, and we can explain it a bit more. So pyruvate is oxidized by the enzymes. And what's the opposite of oxidation? Yeah, so the opposite of oxidation is reduction. And so we know anytime something's undergoing oxidation in our respiration, there will be a coenzyme like NAD involved to be reduced as the opposite. So we have a molecule called pyruvate, which is a 3-carbon molecule, and this is oxidized by enzyme, and it becomes a 2-carbon molecule known as acetate, as it loses 1 carbon molecule as carbon dioxide, and this process is known as decarboxylation. And then this acetate group then combines with the coenzyme A to form acetyl-CoA. And that might seem a bit complex at the moment, but we'll try and go through it step by step if that's okay. So let me clear all my drawings first this time. So we have here a nice diagram that denotes everything we've said so far. So I'll write as we go along. So as you can see here, pyruvate is a 1, 2, 3. It's a 3-carbon molecule. However, it ends up as that's meant to be 2 carbons, but it's a 2-carbon molecule with a CoA attached to it. So it's a 2-carbon molecule. So what happens is oxidation, as we mentioned before. So oxidation is what's happening in the main reaction, whereas the NAD is getting reduced. So as we said before, gaining a hydrogen means reduction. So we lose a hydrogen from our pyruvate group, and we lose a carboxyl group. So if we say our original pyruvate molecule had a carboxyl group, which was our 3rd carbon, it becomes lost as carbon dioxide. And this process is known as decarboxylation because you're getting rid of the carboxyl group in our pyruvate molecule. So it's called decarboxylation. And this is always released as carbon dioxide. And you can see here is the first time we see one of our waste products, which is carbon dioxide, which we release when we breathe out. So carbon dioxide is released when we breathe out, and that's one of the products of our overall reaction as well, which was carbon dioxide plus water. And we get another coenzyme here, and this works kind of similarly to NAD, as it's an enzyme which is not directly involved in the reaction, but it helps the reaction along. And it combines with the acetyl group, which was just 2 carbon molecules, and it joins on to form a acetyl-CoA. So this is a 2 carbon molecule with a CoA group attached. And this is what then goes on to enter the Krebs cycle. Okay. So let me clear all my drawings again. So to summarize what we've learned there, we get summary products of acetyl-CoA, which was our end product of our reaction, which we saw before. We get carbon dioxide, which was our waste product, has no purpose. A reduced NADH, which will come into play in oxidative phosphorylation. And so this is our overall reaction we get. So pyruvate plus NAD plus CoA, which is coenzyme A, makes acetyl coenzyme A, carbon dioxide plus reduced NAD or NADH. So as you can see here, we've lost 1 carbon. So this becomes a 2 carbon molecule. And I need you to remember that this is actually happening twice over per glucose molecule. So because 1 glucose molecule, 1 glucose molecule makes 2 pyruvate molecules, right? So per pyruvate molecules, we're getting 1 acetyl-CoA. So you need to remember this whole link reaction is happening twice. So there's 2 link reactions happening per glucose molecule. So everything we produced here will be doubled in per glucose molecule. So it will be 2 acetyl-CoA, 2 acetyl-CoA, we'll have 2 carbon dioxide, and we'll have 2 reduced NADs. No, reduced NADs. Okay. And this introduced the proportion of the glucose, so the acetyl-CoA, into the Krebs cycle. And that's all that says there. So the coenzyme A is that it helps with the reaction. So we know that the pyruvate undergoes oxidation. So we know the pyruvate undergoes oxidation to become the acetyl-CoA. And the coenzyme just helps along this reaction without directly getting involved. So it changes its rate of reaction almost like a catalyzer, catalate. So this is actually made of a ribose molecule, an adenine, and 1 vitamin, but you don't need to go into any more detail than that. So you just need to know it's made of ribose, adenine, and 1 vitamin. And it's just there to help the reaction go along without getting involved in it. Cool. So it bonds to the remaining, so as we saw before, it bonds to the remaining pyruvate molecule to produce acetyl-CoA, which then delivers the acetyl group to the Krebs cycle, because we'll see how that gets involved, and it keeps the aerobic respiration going. So our next question is, so someone's just asked, what is the role of NAD? So we'll cover that in a bit more detail when we get into oxidative phosphorylation. But at this point, the NAD is just there to help the oxidation of the reaction. So we have the NAD going to NADH, so it's getting reduced, but it's helping the oxidation of these molecules so it can continue in the process of, so for example, in glycolysis, it was getting oxidized from triose phosphate to pyruvate. So the NAD was helping that reaction along. And in this case, it's helping the oxidation of pyruvate to acetyl-CoA. But we'll see why it's important also to have the reduced NADH in a bit as well. So our first question is, what is the net gain of ATP and NADH respectively in this step? Put the poll up. Waiting for a couple more answers. Just take a guess. So I'll end the poll there. So we've had quite a various answers. So I'll explain why the right answer is what it is. So it was a bit of a trick question, actually, just to throw you off a bit. So link reaction, there's actually no involvement of ATP at all. So ATP is not involved in the link reaction. So this is not looking at glycolysis anymore. This is just the link reaction by itself. So there is no ATP involvement in the link reaction. And whereas NADH, you actually do get two of them, because as I said before, you get two lots of the reaction happening. So you get link reaction happening twice over. So if in each one, so in each reaction, you're producing one molecule of NADH, then in per glucose molecule in this step, you'll be producing 2 NADH. So the right answer would be 2. So I could have clarified this question, sorry, by saying per glucose molecule. So per glucose molecule, how much ATP and NADH is produced in the step. So it would be no ATP because we don't actually directly get any ATP from the step. And NADH, there's two lots of them because the process happens twice. And so we get 2 lots of NADH. So the right answer is B, which is 0 ATP and 2 NADH. Okay. So our next question is, by which process is CO2 removed from the pyruvate? Okay, thank you for your answers. So the right answer actually is D, decarboxylation. So we'll look at each one in turn. So dephosphorylation, so if you look at the word itself, it says dephosphorylation. So that means removing a phosphate group. So this was what was happening when we generated ATP. We were removing the phosphate in from the triose phosphate to make ATP. So this did not happen in the link reaction. Dehydrogenation is when we're removing a hydrogen molecule. And a hydrogen molecule, removing a hydrogen. What's another word of saying removing a hydrogen? I've said this. Does anyone know? Removing a hydrogen is, or losing a hydrogen is also known as. So So removing a hydrogen so removing a hydrogen is also known as oxidation, because losing a hydrogen means it's oxidation, and the opposite of that, reduction, would be gaining a hydrogen. Because gaining a hydrogen also means gaining electrons. Sorry, get a eraser. So gaining a hydrogen is reduction, and reduction is also known as gaining electrons as well in chemistry, but we don't necessarily need to go into that. But we'll look into reduction and oxidation in a bit more detail in a bit. And then decarboxylation, which was the right answer, as you can see, carboxyl group. So we're removing a carboxyl group. So there's a carbon involved. So we're getting rid of one in the form of CO2. And that's why this is the correct answer, because we're removing a carboxyl group. So to summarize, the first one, dephosphorylation, is for removing phosphate. Dehydrogenation is removing hydrogen, and it's also known as oxidation. Reduction is gaining hydrogen, and it's the opposite of oxidation, so opposites. And decarboxylation is removing the carboxyl group. I hope that made sense. Cool, we'll go on to the next question. I think that's some more. So how many carbons are there in acetyl-CoA? Sorry, I keep forgetting to run the polls. Okay, I'll end the poll there. So the correct answer was 2. That's because, I'll explain now. So 2 is the correct answer because we have a 3-carbon pyruvate molecule. Yeah, and it's converted into acetyl-CoA. And in this process, we actually lose 1 molecule of carbon dioxide. And so we're losing 1 carbon here. So in that case, we're left with 2 carbon molecules because we've lost 1 here. And so that leaves 2 plus 1 makes 3. So that leaves 2 carbon molecules in the acetyl-CoA. Okay. And then our next question, I think it's our last question with regards to link reaction. By what process does pyruvate enter mitochondria? Sorry, the polls again. Yeah, that's great. Everyone's gone for the right answer, which is active transport. Oops, I forgot to press annotate. So yeah, so the right answer was active transport. And does anyone remember what are the 2 things we need for the pyruvate to get across the mitochondria? Yeah, so we need a small carrier protein and what's the other thing? Yep, a small amount of ATP. And these two things combined make it active transport. That's great. So at this point, does anyone have any questions? I'll relaunch the poll. And if you have no questions and you're fine, press A. If you have any questions or doubts, press B. That's great. Everyone seems to be good. I can't actually see who's voting what, so if you do have any problems, then do. Okay, so I'll do a mini recap of this process, the link reaction with you guys. So I'll get you guys to help me redraw the link reaction. So what was the starting product we had in link reaction? Anyone? What do we start with? Yeah, so pyruvate is the first thing we start with in the link reaction. And does anyone remember what our end product is? What's our end product? So this is our 3-carbon molecule, which gets converted into a 2-carbon molecule called, yeah, acetyl-CoA. Correct. And so this is known as an oxidation process. And as we are becoming from a 3-carbon molecule to a 2-carbon molecule, we undergo something called decarboxylation, which is when we lose one of our carbon molecules as carbon dioxide. And this process is known as decarboxylation. Yeah. And then because our pyruvate's undergoing oxidation, we need something else to be undergoing reduction at the same time, because whenever oxidation happens to one molecule, reduction has to happen to another molecule. And in this case, what we have is our reduced NAD forming. So we have our NAD coenzyme coming in and gaining a hydrogen. So it gains a hydrogen from the pyruvate to make reduced NAD. Also known as reduced NAD. Yeah, that's right. And then how do we get from what we have at the moment is an acetate molecule. So how do we make it an acetyl-CoA? What has to come in to make it acetyl-CoA? Yeah, the coenzyme A. So quite self-explanatory, actually. So there's coenzyme A, which is not actually directly involved in the reaction, but it's just helping the reaction along. And it helps deliver the acetal molecule to the Krebs cycle. And that's how, basically the summary of this reaction. And so this is happening per pyruvate molecule. So per pyruvate molecule, we get 1 acetyl-CoA. How many NADHs do we get per pyruvate? So per pyruvate molecule, we get 1 NADH because it's just one reaction. And we also get 1 CO2 molecule. But because there's 2 pyruvate molecules per glucose, so per glucose molecule, should I write it in a different color? So per glucose molecule, because there's 2 pyruvates per glucose molecule, as we saw before, because you get 2 pyruvates from one glucose molecule, we get 2 acetyl-CoA, 2 NADH, and 2 CO2. So the trouble with exams are they might ask you what are the products per pyruvate molecule, or they might ask you per glucose molecule. So you need to make sure to read the exam question carefully to see what they're asking for, because it's something I found quite difficult because it's quite easy to trip up over. So make sure to read the question carefully and see if it's asking for the glucose molecule or the pyruvate molecule. So in the pyruvate molecule, you get 1 acetyl-CoA, because as you can see here, 1 acetyl-CoA. You get 1 NADH, as you can see here. And you also get your 1 carbon dioxide molecule, as you can see here. But as the whole process is happening twice, this is why you get 2 sets of each of them. I hope that makes sense. Has that clarified everything? I'll run a poll again. So if everything made sense, can you put an A? And if you still have a few more questions, or if you would like me to go over it again, put a B. Yay, everyone's gone to an A. That's great. So now we've come up to 11:45, and our next step in our reaction is the Krebs cycle. And Krebs cycle is quite big, so we might take our lunch break a bit early. And so we'll meet again at 1:45, so at 12:45, if that's okay with everyone. Does that sound good? We'll just take our lunch break a bit early. So I'll meet you guys back here again at 12:45. And then if you have any questions during that, just message me and I'll reply. Hello, I hope everyone's back. So I'm just going to run the poll again to make sure everyone's back again. So if you can just vote for any of the 4 options, so I know you're here. Okay, it seems like most of you are back, so I'll get to move on because we have quite a lot to do in the next session. So the next part of the respiration we'll be focusing on is the Krebs cycle. So clear all the drawings. Okay. So we moved on from the first 2 steps, which was glycolysis, and then our second step, which was the link reaction. And now we'll be going on to Krebs cycle. It's quite complex, so I hope everyone's had enough food and drink to keep them energized for the rest of this. So let's get started. So Krebs cycle, also known as the citric acid cycle, you'll find out why in a second, is a series of reactions where the 2-carbon acetyl-CoA, which I hope you remember, was the product of our link reaction, enters the Krebs cycle. And you can also get acetyl-CoA not only from glucose, you can also get it from fatty acids, which comes from lipid breakdown, and amino acids, which come from protein breakdown. So it doesn't necessarily always have to be glucose, it can be other substrate molecules as well. So these enter the Krebs cycle. And then the acetyl-CoA, which I hope you remember is a 2-carbon molecule, will combine with the 4-carbon oxaloacetate, which is in the cycle itself. And I'll draw it out in a second so it's a bit more clear, to form our 6-carbon citrate. And this is why it's known as the citric acid cycle, because it's got 6 carbons in it. And then the coenzyme A is then released back into the matrix, and then it goes back into the link reaction. So as you can see here, the coenzyme A didn't actually get involved in the reaction at all. It only just helped the acetyl-CoA get to the Krebs cycle, gave the acetyl group to the 4-carbon oxaloacetate to make the citrate, and it's now back on its way to link reaction where it can continue to produce more acetyl-CoA. Acetyl-CoA. So, and then the citrate is again converted back into the oxaloacetate via a lot of oxidation and reduction reactions, so known as redox, so reduction and oxidation, so redox reaction. And we'll look into it in a little bit more detail now. So let's go on to the next slide. So let's get a blank slide up. Okay. So to start off with, the molecule we started off with was, and does anyone remember what that 4-carbon molecule was called? Don't worry about the spelling. Yeah, so it was called oxaloacetate. So oxaloacetate is the 4-carbon molecule that we start with. And then it gets converted into a 6-carbon molecule. Sorry, I think I started a bit too far to the left side. So let me just erase that, start again. So oxaloacetate is our 4-carbon molecule. It then is converted into citrate. How many carbons does citrate have? Can anyone remember from the previous slide? Anyone? So citrate gets converted, is a 6-carbon molecule. And does anyone remember what was the product of the link reaction, which joins with the oxaloacetate to form citrate? What was the product of the link reaction? Yeah, so the product of the link reaction, if you remember, was acetyl-CoA. Acetyl-CoA. So what happens here is, as you can see here, it's a 2-carbon molecule and a CoA enzyme. Yeah, so if I was to draw it out, say this was my carbon and this was my carbon, and it's attached to a CoA molecule. So these 2 carbons join onto the oxaloacetate group. So the acetyl portion of acetyl-CoA joins onto oxaloacetate to make citrate. And this CoA enzyme is actually released back into the link reaction. I'm just moving this so I can see better. Okay, so the CoA enzyme is released back into the link reaction because it doesn't actually get involved in any of the processes. So it donates the acetyl group, which is 2 carbons. You can see it, 2 carbons. It adds onto the 4 carbons to make it 6-carbon molecule. And the CoA enzyme goes back into the link reaction to make more acetyl-CoA, and which then can continue the Krebs cycle again and again and again. And then this citrate molecule is converted back into oxaloacetate in quite a few steps. So we'll break this down bit by bit. So first of all, we get it being broken down into a 5-carbon molecule. And we don't actually need to know the name of it at this stage because there's a lot of steps into it. So we just need to know it gets converted into a 5-carbon compound. Does anyone remember what process things go under if it's losing a carbon? Does anyone remember what the name of the process was when you lose a carbon? Yeah, it was called decarboxylation. So in decarboxylation, so that's actually what's happening here. So what we said before was in decarboxylation, we lose things in the form of CO2, right? So we release CO2. So here we lose a CO2. And I'm actually just going to start a table here of all the products we're getting out of this reaction. So let me start one here. So first of all, we have CO2. I'm going to start a tally here. So we have 1 CO2 so far. And then at the same time, we actually have oxidation happening while the decarboxylation is happening. And what coenzyme usually comes in when there's oxidation happening? Does anyone remember? Yeah, NAD. So NAD gets involved. And so NAD comes in and is reduced to form NADH. So NAD plus becomes reduced NAD or NADH. So this process is known as dehydrogenation. So we said, so it can be also known as oxidation, but in this step we call it dehydrogenation because what's happening here is the citrate is also losing a hydrogen. So you're releasing a hydrogen, which then gets donated to the NADH. So if I was to write out the equation here, it would be NAD plus H plus makes NADH. And just to balance the equation, I'll put the 2 electrons in there as well. As you can see here, the NAD combines with a hydrogen from the citrate to form reduced NAD, also known as NADH. So if we're going to add it to our tally, we have another NADH here as one of our products. Okay, and then moving on from here, we go into the next step in a series of steps. So this whole step actually happens in multiple steps. And although you don't need to know each step individually, I'm just going to break it down for you just to make it a bit easier to understand. So I'm just going to change the color of the thing just to show you that you don't need to actually remember each step, just so that you can have it in your memory, just to make it a bit easier for you to understand, really. So what happens first is it gets converted into a 4-carbon molecule. So what actually happens here is the same process as what you just saw before. So can anyone tell me what are the 2 processes that happens here? So you get decarboxylation and oxidation. So yeah, these 2 processes happen again. So you get your decarboxylation and dehydrogenation. So you release another molecule of CO2 and another molecule of NADH. So I'm going to add that to our tally here. So we get another molecule of CO2 and another molecule of NADH. In the next reaction, what actually happens is the 4-carbon molecule rearranges itself. So can you get the pen back? So in the next step, the 4-carbon molecule just rearranges itself. So there's no losing of carbon. So we don't undergo any decarboxylation. It just rearranges itself. But actually in this process, because it's trying to rearrange itself, we need some, we actually form some ATP in this process, because a molecule of ADP comes up and then combines with the inorganic phosphate, which is attached to this 4-carbon molecule, and then forms ATP. Okay, and in this process, you also release dehydrogenation also happens, so you form some NADH. So all this happens here is you get something called substrate-linked phosphorylation. So I'll write that down in a nicer color because I feel like it's a bit hard to see. So substrate-linked phosphorylation. So the ATP here is generated by substrate-linked phosphorylation. And all that means is it's linked to this substrate and then it combines with a phosphate, so it's phosphorylated to form an ATP. So while the 4-carbon molecule is rearranging itself, it releases an inorganic phosphate, which combines with an ADP molecule to form ATP. And in this process, dehydrogenation also occurs again, and you get another molecule of NADH. So adding to our tally now, we have a molecule of ATP and another molecule of NADH. Okay. And then in our last step, and there's actually more steps if you go in to do it at uni, but this is just to make it easier for you guys to understand. What actually happens here is it rearranges itself again. But this time, instead of NADH being involved in the dehydrogenation process, we get another coenzyme called FAD+. And this goes on to make FADH2. So just to balance the equation, we get 2 hydrogen pluses and 2 electrons. So this works the exact same way as as NADH, but it's a better molecule for this part of the reaction. So what we get released here is an FADH2 molecule. It's exactly the same process as the NADH, but it's just a different molecule is more suitable at this stage. So adding to our tally, we now have FADH2 as well, which is 1 molecule. Okay, so I hope that's all made sense, and I'll just go through it all again, well, now that I'm not drawing it out. So let's pick a different color. So first of all, you have your oxaloacetate, which is a 4-carbon molecule, which combines with your 2-carbon acetyl-CoA And the CoA, which is not actually involved in any reaction, goes back into the link reaction, where it goes on to make more acetyl CoA, like we just saw before in our previous, in the previous part of our presentation. The oxaloacetate, now that it's combined with the 2C of the acetate group, it becomes citrate, which is 6-carbon molecule. And then it is decarboxylated to form a 5-carbon molecule via 2 processes. So you guys said decarboxylation is one, where it releases it in the form of CO2. So we release some CO2 there. And dehydrogenation, where you release a hydrogen. And so you get NAD becoming NADH. And that's how you got one of your carbon dioxide molecules and one of your NADH molecules. This happens again as you get your 4-carbon molecule from your 5-carbon molecules. So you release 1 molecule of CO2 and you release 1 molecule of NADH. Should have done that. Okay. And then what happens is just a lot of rearranging of the 4-carbon molecule because it's slightly unstable. So first of all, we get a substrate-linked phosphorylation, which is the name given to how this ATP here is generated. So the ADP combines with an inorganic phosphate in this reaction, and it goes on to form 1 ATP, and that's how you got your 1 ATP molecule. And dehydrogenation also takes place during this rearrangement, and that's how you got your 1 NADH molecule. Okay, and then in our last step, when it's rearranging itself again, then you get your FADH2 molecule, which acts the same way as NADH, but it's just a more, that coenzyme is more suitable for that step than previously. I hope that's all made sense. So, and then just to expand on how the ADP forms, we said, as we said before, ADP plus pi inorganic phosphate makes ATP. So that's your ATP molecule combined to your 2 phosphate molecules, and then you add another phosphate molecule to it to make it a 3-phosphate molecule. I hope that's all made sense. And I just quickly run a poll to just make sure everyone's on the same page. I'm happy to go over it again if you want me to as well. That's great, thank you. Just going to end the poll. And just one last time, I'm just going to go over it because I just want to make sure you guys all understand, because it's probably one of the hardest parts of Krebs respiration, and the part I found the hardest to understand when I was doing A-levels. So first, starting off with the oxaloacetate, which is the 4-carbon molecule, combining with the 2-carbon acetyl CoA. So I have a question which said, where did the FAD come from? So much like the NAD, these are just coenzymes floating around in the matrix, and it just joins in because it's just more suitable for that step of the reaction. So the NAD also just hangs around in the matrix, and this gets regenerated later on, and I'll show you when that happens as well. So the oxaloacetate combines with the 4-carbon molecule, combines with the 2-carbon molecule to make citrate, which is the 6-carbon molecule, which then is decarboxylated to a 5-carbon molecule. Then it's again decarboxylated to a 4-carbon molecule, and then it's returned to its oxaloacetate site. So I've broken down these 3 steps here, but you don't actually need to learn it in 3 steps because you just need to know all the products that come out of it. So if I just clear all my drawings now, and we'll go on to the next slide. So as you can see here, it's a simplified version of what we were just looking at. So the oxalic acetate, which is our 4-carbon molecule, combines with acetyl-CoA, and the coenzyme A is released back into the link reaction. And then this becomes a 6-carbon molecule because it's a 4-carbon plus a 2-carbon molecule makes a 6-carbon molecule. Then we get decarboxylation as it releases a CO2 molecule here. And then we get dehydrogenation. Here as NAD is reduced to NADH, and we release a hydrogen from our citrate. And this is actually reduced here. This part is reduced and this part is being oxidized. So our NADH is reduced and our citrate is oxidized. And then this formed our intermediate carbon of compound of 5-carbon molecule because we lost a carbon dioxide molecule. Then this is again decarboxylated to release a CO2 molecule. So it returns it back to a 4-carbon molecule. It undergoes reduction again. It undergoes dehydrogenation again in the form of creating NADH. And then also undergoes some substrate linked phosphorylation as it rearranges itself. Phosphorylation. And then we get some reduction of the FADH molecules as well. And we also again have the reduction of NADH molecule. So in total, you can see we get 2 carbon dioxide molecules. We get 3 NADH molecules, also known as reduced NAD. We get 1 ATP molecule and 1 FADH2 molecule. And this is also known as reduced FAD. But please remember, this whole cycle actually happens twice per glucose molecule because we had 2 sets of pyruvates, which made 2 sets of acetyl-CoA, which then goes on to do 2 sets of Krebs cycle. So this is your products of 1 Krebs cycle. 1 Krebs cycle. And if someone asked you, if the exam question was, what are the products of the Krebs cycle from a molecule of glucose, you need to make sure that you're doubling everything by 2, because then you'd have accounted for the 2 times the Krebs cycles running per Krebs cycle, per glucose molecule, sorry. I hope that made sense. So, clearing all my drinks again. Okay, great. Now we'll go on to the next slide. So as you can see, the acetyl-CoA from the link reaction, so this is just a summary of what we've drawn out. The acetyl-CoA from the link reaction and the regeneration of the oxalic acetate. So that's a cycle and you can't count them as products. Whereas the products of this reaction is the reduced NAD, the reduced FAD, and the ATP. So as we said before, we get 3 per Krebs cycle, 1 per Krebs cycle, and 1 per Krebs cycle of ATP. And then you times everything by 2 per molecule of glucose, you get 6 per glucose molecule. 2 per glucose molecule and 2 ATP per glucose molecule. But this is per Krebs cycle, 1 Krebs cycle, 1 Krebs cycle. Okay, so things like oxaloacetate and citrate cannot be counted as products because they stay inside the cycle and they keep running the cycle again and again and again. Whereas the reduced NAD, the reduced FAD, and the ATP come out and then go on to the next step of respiration. Okay. So the oxaloacetate can also be known as the acceptor molecule, as it accepts the acetyl-CoA and then it continues to be regenerated. So simply by adding another acetyl-CoA, so if you have more glucose, so when you're consuming food, when you have more and more glucose, you produce more acetyl-CoA and the cycle can keep running as long as you have oxygen, of course. So, and then it's regenerated by redox reactions. So what product is our waste gas in decarboxylation? Does anyone remember? Yeah, it's CO2. CO2 is our waste gas, and we get 2 per cycle and 4 per glucose molecule. So this is just again summarizing what we went through. So the oxaloacetate is regenerated in a series of processes where we have reduced coenzymes NAD and FAD, which gains the hydrogen atoms from the citrate molecule and the 5 carbon molecules to become reduced, and it becomes 3 NADH and 1 FADH2. Okay. Sorry. Okay, moving on. So then the step we had in our cycle was the substrate linked phosphorylation. This is when we had the ATP being generated. So we had our ADP molecule, which looks something like this. It combines with an inorganic phosphate molecule to form ATP. Which is three phosphate molecules as opposed to the 2. And the phosphate comes from the cycle, the inorganic phosphate. Okay. So this is just breaking down the steps of what we covered when we drew out the cycle before. So just a quick question, and I'm going to release my polls again. How many molecules of NADH are produced in this cycle per glucose molecule? So see that it says per glucose molecule. That's great, thank you. So we have the split of answers, so we'll go through it again. So what we learned from before was, I can find my pen. So the correct answer was D, and this is because it's asking you per 1 molecule of glucose. So as I said, 1 Krebs cycle makes 3 NADH. Yeah, but per glucose molecule, what we actually having? So per glucose molecule, we get 2 pyruvates, as we learned in glycolysis. We also get 2 acetyl-CoA because each pyruvate makes 1 acetyl-CoA. And then because of 2 acetyl-CoA, we get 2 Krebs cycles. Yeah, so because of the 2 Krebs cycles, we need to double the NADH. So we need to times it by 2, which gives us 6. Does that make sense? Okay, so I'll clear my drawings again and go on to the next question. So this was just a trick question because exams do like asking questions like this. So you need to make sure, are you answering, is it per Krebs cycle or per molecule of glucose? So our next question is, what is the name of the 4-carbon molecule in Krebs cycle? So let me get the polls up again. Okay, so we have a split of answers again, so we'll go through it again. So I hope you'll remember that initially when we start off with pyruvate in our link reaction has 3 carbon molecules, and then that gets decarboxylated to acetyl-CoA. So we actually release the CO2 there. And so because we lost a carbon, this was actually a 2-carbon molecule. So this is a 2-carbon molecule. Oxaloacetate, on the other hand, is a 4-carbon molecule. And then this combines with the acetyl-CoA to make the 6-carbon molecule, which is citrate. So if we were just to go back to our drawings from before, so as you can see here, we have our 4-carbon molecule, which is oxaloacetate. So you can see 4 carbon molecules here. It combines with a 2-carbon molecule, which is acetyl-CoA, and then the coenzyme A is released back into the link reaction. These two, the 4-carbon plus the 2-carbon, make citrate, which is the 6-carbon molecule. This then goes on to form a 5-carbon intermediate compound, which we don't need to know the name of, which is quite good. And then we go back into oxaloacetate again, which is a 4-carbon molecule. And this is a quite easy 1-marker question they can throw you in because you're meant to know the process of Krebs cycle. So oxaloacetate is your 4-carbon molecule. The acetyl coenzyme A is a 2-carbon molecule. Your citrate is a 6-carbon molecule. And then going back here, you get your 1 NADH formed in your first dehydrogenation as it goes from a 6-carbon to a 5-carbon molecule. You get your second and your third NADH as it comes from a 5C to a 4C. So you get your second and third one. That's your 1, your 2, and your 3. But because this whole cycle happens twice per glucose molecule, we say there's 6 in total per glucose molecule. And then, as you can see here, there's 1 FADH molecule per cycle, but because it happens twice, you say there's 2 FADH2 molecules. And then there's one ATP here per cycle, but because it's happening twice again, we say there's 2 ATP molecules in total. And this is per glucose molecule. Okay, so I'm just going to run a quick poll to make sure everyone's still on board with that. So please put yes, A, if everything's okay and you're good, and B, if you still would like more clarification. That's great. Everyone said it's fine. So we'll go on to the next part, which is oxidative phosphorylation. But before that, because we've done quite a bit now, does everyone want a 5-minute break, or are people happy to go on? I'll do a poll for that as well. So if you want a 5-minute break, put A. If you want to go on, then please put B. Ooh, we have a split. So we'll have a very quick, like 2-minute break, just so that people can get a breath of fresh air before we continue with oxidative phosphorylation. So we'll start in at 25 past, okay? Okay, I hope everyone's back from the very short break. I'm just giving you frequent breaks because one of my lecturers once told me we only take in information in bite-sized pieces. So you're meant to give yourself breaks like every 20 to 25 minutes. So that helps you retain it better. So can you vote again if you're all back, please? Any of the options will do for the poll. That's great. We all have everyone back. So we'll move on to the final step of respiration. But before that, we'll look into a bit more detail about the coenzymes NAD and FAD. So they are coenzymes, much like coenzyme A, and they don't get involved in the reaction itself, but they facilitate the reactions. So they play a critical role because they accept hydrogen atoms. And what did I say was the process of gaining hydrogen called? Can someone pop it in the chat? Yeah, so this process is known as reduction. So this is known as reduction. And this is very important because all these reduced NADs and FADs come into use later on in the reaction in our next step, which is oxidative phosphorylation. So the hydrogen atoms it gains contains a hydrogen ion, so one proton and one electron, as you can see in this diagram on the right side. So as we said just now, gaining a hydrogen is known as reduction. And if you've done chemistry, you'll also know reduction is also known as gaining electrons. And then when hydrogen is removed, it's known as oxidation. And this is also known as losing electrons. So a nice mnemonic that I've used since I was in high school is oxidation is loss and reduction is gain, so oil rig. So you can see the reactions that are happening here with our coenzymes. So NADH plus the 2 NAD plus the 2H plus makes our NADH plus H plus. So when it's becoming reduced NAD, so it gains the hydrogen, this is reduction. And when it goes the opposite way and it loses the hydrogen, this is oxidation. It's the same for FAD, so they're very similar molecules. It's just at different stages of our Krebs cycle, we use different ones. So FAD, when it gains a hydrogen, it becomes FADH2. And when it loses the hydrogen, it becomes FAD plus 2H. Okay. So they're very important because they play a role in the electron transport chain. And we'll go into a bit more detail about what is that when we look at oxidative phosphorylation. So they're very important, these electron transport chains, in the synthesis of ATP, because electrons are released from reduced NADH, because you probably saw when we are reducing NAD, so the equation would be NAD plus H plus makes NADH. But to balance that equation, we need an electron. So as you can imagine, when the opposite is happening, when this reduced NADH is oxidized, you release the NAD plus the H plus electrons. And so these electrons that are released are involved in the electron transport chain, which we'll go into a bit more detail. And the hydrogen ions are also released, which are also important in the electron transport chain. And so this is the same with the FADH2. So FADH2 also releases its electrons, releases 2 hydrogens and 2 electrons. So as you can see here, they both release electrons, which are then used in the electron transport chain. And we'll go into the importance of the electron transport chain in a bit. So the electron transport chain pushes hydrogen ions across the inner mitochondrial membrane. So if I can just draw a tiny diagram right here. So say this is our outer membrane. This is our highly folded inner membrane. It's our inner membrane. So our matrix on this side and our intermembrane space, so IMS here. So what happens is the electron transport chains, which are like carrier proteins which sit on this membrane, they push H plus ions into the intermembrane space using a proton pump. These protein transporters are called protein pumps because energy is released when the electrons released from the NADH. So what happens is the NADH, as we saw before, releases the NAD plus the electrons, and these electrons then go into these transport chains and release some energy, move on to the next protein, release some energy, move on to the next protein, and they keep moving down the chain. And that's why it's known as an electron transport chain. And the energy released by these electrons are then used to pump these hydrogen ions into the intermembrane space. So there's a very high concentration of H plus in the IMS. And what does this mean for the pH? If there's a high concentration of H plus in the IMS, what is the pH in the intermembrane space? Is it high or low? Yeah, so it's low because there's a high concentration of H plus, which is acidic, then this means there's a low pH. And that's why at the start, when we were looking at the structure of the mitochondria, we said the intermembrane space has a low pH because it has all these hydrogen ions being pumped into it. This naturally means there's a gradient down, and then the H ions wants to travel down that gradient, and that's when ATP is synthesized. But we'll go into a bit more detail instead of this tiny diagram in the corner in a bit. So oxidative phosphorylation is the last stage of aerobic respiration. I'm sure you'll be all glad to know. It occurs in the inner mitochondrial membrane. So as we remember, glycolysis happens in the cytoplasm. Link reaction and the Krebs cycles takes place in the matrix, whereas oxidative phosphorylation happens in the inner mitochondrial membrane. And this is where we produce most of our ATP molecules. And this is why it's so much better than anaerobic respiration, because that only takes place outside the cell. We're not generating a lot of ATP. And this is also where we form our water from the oxygen. And this term here, the oxygen is a final electron acceptor, or you can also use terminal instead of final if you want to. Terminal electron acceptor is a phrase that exams really like. So make sure that, and whenever you're mentioning oxygen in your exams, you like to say it's a final electron acceptor, because that's usually a mark in the exam. So it combines with the protons, also known as the H plus ions, and the electrons to make water at the end of the electron transport. We'll draw this out in the next slide. So maybe in a couple of slides. So this is just going back to the Krebs cycle. So as we know, at every stage, there's a doubling of the reduced NAD because there's one glucose molecules, but two pyruvates and two acetyl-CoAs, which means two Krebs cycles. So this is just summarizing what I told you guys before. So for one cycle, there is three NADs in the Krebs cycle, but when you, just getting my pen. So we formed two NADs in glycolysis because it's one per reaction, but it's two per glucose molecule. So this right side is per reaction, and this is per glucose molecule. Sorry. This is per glucose molecule. glucose molecule, because there's a doubling effect. There's 2 formed from the link reaction, and you get 6 because there's 1 which doubles to 2, 3 which double to 6 in the Krebs cycle, and then 1 FAD in 1 cycle of Krebs cycle which doubles to 2 per glucose molecule. So everything you just double to 2 because the glucose is split into 2 molecules of 3. So because of this, all of these NAD and FADH, so if you add them all up, there's 8 NADHs. That's not 8, I can't add. 10 NADHs and your 2 FADH2s, they all enter oxidative phosphorylation and they're all involved. And you can see why that would lead to lots of ATP when they're oxidized. So it's phosphorylation. So in our next slide, hopefully we'll be able to draw it out. Okay, so I'm just going to take a second to draw out our inner membrane and outer membrane. So we have our outer membrane here. Let's label that outer membrane. Outer membrane. Then we have our highly folded inner membrane. So inner membrane. Between here, we have our intermembrane space. And does anyone remember what's on this side of the inner membrane? Can you put your answers in the chat, please? So if the intermembrane space is here, what's the solution which is on this side? The aqueous solution that contains the enzymes and DNA, where our Krebs cycle and things takes place. Yeah, so it's the matrix on this side. So if you remember, the Krebs cycle takes place in the matrix. So all our reduced NADHs and FADHs would be produced here. Okay. Then I'm just going to draw our electron transport chain, maybe in a different color. So this is our electron transport chain. And then we have our ATP synthase here. ATP synthase. And I'll show you why it's important in a bit. So this is our electron transport chain, ETC. So what happens is when NADH is oxidized, it releases NAD plus E minus, electron, sorry, H plus. So as you can see, it releases a electron, 2 electrons, and a hydrogen ion. So these electrons actually go on to release some energy to these carrier proteins. Then they go on to the next one, release some less energy, go on to the next one, release some more energy, and then go and release some more into this one. And then finally, what happens is, if you remember, we had glucose and oxygen as our starting substrates for our respiration reaction. So oxygen comes here, and oxygen combines with our electrons, and we'll balance the equation in a bit to make our H2O, which is our other product of respiration, which is water. So as you can see, the oxygen is called the final electron acceptor because that's exactly what it does. It's the final thing which accepts the electrons. It also has, so this energy which the electrons release as it goes from one to the other, releases a tiny bit of energy. And what that does is that's enough energy for these H+ ions, which are being released alongside the electrons when they're oxidized. They're pumped into the intermembrane space using the energy from the electrons. So it's not energy from ATP, it's energy from these electrons that are moving from transporter to transporter. So because they're all acting to pump the hydrogen ions into the space, you get a lot of H+ in here. If I put in a lot of H+s. Okay. And then because there's so much H+ in here and very little H+ in here, because they're all being pumped to the side, the H+ ions want to move back in to the matrix down the concentration gradient. So this is known as the chemiosmosis theory, as it wants to move down the chemical gradient. But it can't, hydrogen ions, because they're ions, they can't pass directly through the inner membrane. So it needs this protein called ATP synthase. And do you guys know what process it's called if you're using a transport protein to move down a concentration gradient? What's that transport called? If you're using a transport protein, a carrier protein to move down a concentration gradient, what's that type of transport called? Yeah, facilitated diffusion. So that's exactly what happens here. So the H+ ions are facilitated, so they go through facilitated diffusion to get into the matrix via the ATP synthase molecule. So as you'll remember from the start of our lecture, ATP synthase is the protein which synthesizes ATP. So as the H+ ions are coming in, a reaction happens where ADP joins onto an inorganic phosphate molecule, which is on the carrier transport to make an ATP molecule. Sorry, that's just illustrated really badly there. I'll draw it in a different color to make it a bit more emphasized. So an ADP molecule comes along. It reacts with an inorganic phosphate, which can be found from the shape changing in the facilitated diffusion, which releases it. So as you might have learned when you learned process of transport, when the shape of the facilitated diffusion carrier proteins change, they might release an inorganic phosphate. So the ADP binds onto that to make your ATP. And because there's so many ions on this side, so for every single NADH you had reduced and every single FADH you had reduced previously in your Krebs cycle and your link reaction and your glycolysis, they're all coming here and being released as electrons and H+ ions. So because there's so many H+ ions going into here, you can generate a lot of ATP. And this is the main area where our ATP is generated. So coming back to our oxygen, which is our final electron acceptor, we can see here it's accepting the electrons, but because there's hydrogen ions here as well, we combine with the hydrogen ions as well. So you get to make it a balanced reaction, so you combine it 2 electrons and 2 hydrogen ions to make H2O. And so this is our final balanced reaction, where our H2O is produced as our waste product. So we saw carbon dioxide being produced in both the link reaction and the Krebs cycle, but this is where water is produced, so that we get glucose plus O2 makes CO2 plus water. Okay, so you can see where these products come from. I understand that I've gone through it quite fast. But I'll just run a poll to see if everyone's understood or if they want me to go through it again. So if you want to say A, if you understood everything, and B, if you want me to go through it again. That's great, everyone's understood it. So just cross this off and then clear all my drawings again. So this is just summarizing what we've drawn out. So what we saw there was the chemiosmotic theory, because the hydrogen ions were first pumped against their concentration gradient into the intermembrane space using the energy from the electrons. So just 1 second. So using the energy from the electrons. Energy from electrons. And then we also had, but then it moves back down the concentration gradient via facilitated diffusion via the ATP synthase channel. And this allows the change in shape of our carrier protein, allows the ADP to combine with an inorganic phosphate to form an ATP molecule. And this is just, again, summarizing what we've discussed so far. So the reduced NADH and the reduced FADH donate the hydrogen ions, which split to give the ions and the electrons, which then go on to act in the electron transport chain and release energy. Okay, so they're all tightly packed because, as we said before, the cristae are highly folded, so a lot of electrons can flow between them easily. And the inner membrane is impermeable to hydrogen ions, and that's why we need the facilitated diffusion of the ATP synthase. So this here is just a summary of what we've drawn out. So we get the oxidation of our NADH and FADH, which releases the H+ ions, and the electrons, which move down the transport chain, releasing energy. And then these H+ ions are pumped against their concentration gradient via these proton pumps. They then travel down the concentration gradient via facilitated diffusion. And then this allows the change in shape of the protein to combine ADP and the inorganic phosphate to make ATP. I hope that's made sense. And then the hydrogen ions and the electrons all combine to make water, which is just everything we've discussed so far, just in an image, so you can go over it later if you want to. So why do we need oxygen? So it is the ultimate electron acceptor. So I'm just going to keep repeating that because you need to mention that in your exam. And the electron transport cycle needs oxygen, otherwise electrons have nowhere to go. Because if the electrons can't move down the chain and then get accepted by the oxygen, they're going to get stuck, and then they're not going to move down that chain, which means then the NADH is not going to oxidize more. Then there's a buildup of NADH, and then our whole cycle stops. So that's why we need oxygen, because that oxygen accepting all those electrons at the end is what's keeping this whole chain going. If there's none of that, the electrons can't move and the NADH can't be oxidized, and so the whole cycle stops. So we have some more questions here, and I'll restart my poll. Where are H ions moved to by the proton pump? So where does the proton pump the hydrogen ions to? Yeah, that's right. So I think everyone's understood. So the answer was intermembrane space, and then this is where it's pumped to, whereas the matrix is where it comes back into via facilitated diffusion. Cytoplasm is outside the mitochondria, so it can't be that. And the inner membrane itself is impermeable to the proton pump, so it can't move into the membrane. Cool. Next question. Let me get rid of my drawings. Okay, our second question is, by what process does the H plus re-enter the matrix? Yep, so most of you got the right answer. It was facilitated diffusion, because remember, it comes down the concentration gradient. So it comes down the concentration gradient via ATP synthase. So we're not using up ATP, we're actually generating ATP synthase. We're generating ATP, not using up ATP, so it can't be active transport. And active transport is also against the concentration gradient. Simple diffusion means it won't need a protein, but we know the H plus is impermeable to the membrane, so that it has to have a protein. And then it's not co-transport because something else needs to go alongside it, but it's a facilitated diffusion. Okay. Clear my drawings again. Ooh. So our next question is a nice easy one. Which enzyme is involved in the synthesis of ATP? Yeah, that's right. It's ATP synthase, and I won't bother drawing on this one. I feel like I've told you guys enough. ATPase is actually the enzyme which breaks down ATP and which won't be involved here. And aldolase is just another random enzyme that I decided to put in. Okay, so anaerobic respiration, on the other hand, is when we have no oxygen. So none of our link reaction, our Krebs cycle, and oxidative phosphorylation will not happen. And so there's no oxygen, so there's no final acceptor of electrons. So the only process of our normal respiration that happens is the glycolysis part. Yeah, so as we said before, the reduced NAD, FAD aren't oxidized, then the Krebs cycle stops and the link reaction stops because acetyl-CoA is not used up. So the whole thing stops apart from glycolysis. So we can generate and it's some ATP via anaerobic respiration, but it's very inefficient compared to the whole respiration process, because the only thing it can generate ATP through is glycolysis. So does anyone know what fermentation it is called in yeast and microorganisms in anaerobic respiration? So it's actually called ethanol fermentation. And as you might know, we put yeast in stuff to produce ethanol. And so that's actually a useful product, say, in making beer and things. And so that's how you get ethanol fermentation. But in humans, we actually have lactate fermentation. So I'm sure you all know that when we run for ages, we get lactic acid buildup in our muscles. And that's because when we run out of oxygen, then our body starts to produce lactate instead because it's doing it via anaerobic respiration. And so that builds up as an acid in our muscles, and the acid causes denaturing of enzymes, which can then cause pain. So in ethanol fermentation, what happens is the reduced NAD transfers its hydrogen to ethanol to form ethanol. And the pyruvate is decarboxylated to ethanol in the first step, producing carbon dioxide. And then the ethanol is reduced to ethanol by an enzyme, alcohol dehydrogenase, which acts as an hydrogen acceptor. So we'll look at that in here. So first of all, we have our decarboxylation. Our decarboxylation, which is happening here. Decarboxylation, as we release a CO2 molecule. So we lose a carbon and we're left with CH3, CHO. And our end product is CH3, CH2OH. As we can see, we gain a hydrogen molecule here. We gain two hydrogen atoms. So that comes from the oxidation of NADH, which was produced in glycolysis. So if you remember, we gained two NADHs from glycolysis. They undergo oxidation and they release this hydrogen, and this forms ethanol, and that regenerates the NAD, which then you can use in glycolysis again to produce more pyruvate. And you can generate that ATP in that process, and you can keep that cycle going. But as you can see, that only produces 2 ATP per glucose molecule. And so that's not very efficient compared to all the ATP we can produce by aerobic respiration. So just going on to how we do it in humans now. So this is a one-step process, so it's slightly easier. So the enzyme is lactate dehydrogenase, whereas last time it was alcohol dehydrogenase. And this is just one step. So again, the NADH is oxidized to NAD and releases the hydrogen, and the pyruvate is converted into lactate. And the NAD, which is then regenerated, can then go back into glucose and take part in glycolysis, as we saw before, and generate more ATP. But this is not necessarily efficient because we're not generating a lot of ATP here. So this is just repeating what we just said. It's oxidized back to pyruvates and moved into the Krebs cycle. And then we might need, because it's acidic and then because it's denaturing to enzymes, we need extra oxygen to compensate for it when we start breathing again. And so that's why we breathe deeper and faster after exercise, to compensate for the oxygen debt we created in that time. And then lactate can also be oxidized, converted into glycogen for storage in the liver. And that's the two ways lactate can be converted. So it can be oxidized back to pyruvate to enter the Krebs cycle, or it can be converted into glycogen for storage. So before we go on to the exam questions, because that was a lot of stuff we covered, should we have a 15-minute break or 10 minutes breaks? Shall I put up a poll again? If you would like a 10-minute break, press A. If you'd like a 15-minute break, press B. Actually, no, 5-minute break, A, 10-minute break, B, 15-minute break, C. So most of you have gone for a 10-minute break. So we'll meet up here in 10 minutes, so at 10 past. And then we'll go through some exam questions. And if you have any questions in the meantime, feel free to pop it on the chat. If you're back, can I get you to put on the poll, please? Okay, so it's 10 past, so I hope everyone's back. So I didn't realize we were meant to finish at 2, so we'll quickly go through some exam questions and try and wrap up as soon as possible. So the first question that I was actually going to do was where the respiration takes place and the steps, but I'm sure you guys know that by now, so I'll just skip over it. So the first question we're going to actually focus on is the formation of acetyl-CoA in the link reaction. So if you just think about it, and then we'll take it step by step. So the first thing we need to always say is, what do we start with? So we start with our pyruvate molecule. So our pyruvate molecule is converted into acetyl-CoA. And does anyone remember what the process is called? Yeah, decarboxylation. So decarboxylation happens to remove the CO2. So you say CO2 is released. And also what's happening is oxidation is happening. So the pyruvate's actually oxidized to form acetate, And then because you're saying this one's for oxidized, you need to always complement it with what is reduced. So in this case, what was the molecule that was reduced? What coenzyme gets reduced in the link reaction? Yeah, so you also need to say NAD. So that's your second mark. So NAD is reduced to form reduced NAD. Or you can say NADH, whichever. And then your third mark comes from saying the coenzyme A is added. So it's a 3-step process. So pyruvate is oxidized, first of all, and then CO2 is released in the process of decarboxylation. And secondly, your NAD is reduced to form reduced NAD. And then thirdly, our coenzyme A is added, and that would give you 3 marks. So when you look at exam questions, make sure to look at the amount of marks you're getting for them, because you can try and aim for 1 point per mark. So pyruvate's oxidation would be 1 mark, followed by NAD being reduced to form reduced NAD, that's your second mark, and coenzyme A being added is your third mark. Okay, and your next question. So our next question is, fumarate is a step in our Krebs cycle, and it's explain how fumarate enables the water to then bind to the enzyme. So at first you might think, oh, we haven't covered this, but what you need to look at it separately is that this is actually asking you an enzyme question rather than a Krebs cycle related question. So it's asking you just the generic question of binding to its active site. So you say the fumarate binds to the active site of the fumarase. The fumarase, which changes the shape of the active, which changes its shape a bit, which means it can now, bind. Now it can bind to the water molecule. So I think although this might not be the exact exam answer, what I was trying to show here is that you need to make sure that you're not surprised by the question. So for example, if you see something like this and then you think, oh, these are words I've never seen before, the point of putting them in is just to make you think of the processes itself. So so your marks would come from saying the fumarate binds to the active site of the fumarase, and then it changes the shape of the active site. So it molds itself around it. And so that it can now bind to the water because it's a complementary shape. It's a complementary active site. It is now complementary. So your mark would be from saying the word complementary. So try and not get frightened basically by questions that have different molecules in, because these are just steps within the Krebs cycle, but you don't need to know it to answer the question. You just need to know the basic biochemistry behind it. And then for our next question, is asking for the mechanism by which ATP is formed in the mitochondria. So the main thing I wanted to highlight for this is that we can't talk about glycolysis. No mention of glycolysis. This is because glycolysis occurs in the cytoplasm, and this question is specifically asking about how ATP is formed inside the mitochondria. So we can't mention about the ATP being formed during glycolysis, but you can say ATP is generated during Krebs cycle. Krebs. And then the rest of your marks would actually come from the you explaining the rest of the oxidative phosphorylation process. So you would talk about how Krebs cycle or your link reaction or glycolysis forms reduced NAD and reduced NAD and FAD. And then you would talk about how they're oxidized to release electrons. And then you would talk about the electron transport chain and H+ ions being pumped across the concentration gradient. Then you would talk about, you need to mention, like, you always get a mark for saying oxygen is the final electron transporter. And then you need to definitely mention, because none of this so far says how the ATP is actually formed, you need to talk about how facilitated diffusion happens. Diffusion happens across ATP synthase. Across ATP synthase, and then the joining of the ADP plus inorganic phosphate in this process to make ATP. So what I'm trying to summarize here, although it's not the most elaborately written exam question answer, is that you can't mention glycolysis as generating ATP, although we know that does generate ATP. This is specifically asking about the mitochondria. So we can talk about the ATP that's generated in Krebs. Then we need to talk about the reduced NAD and FAD, which is then oxidized to release the electrons, which then release energy in the electron transport train, which pumps H+ ions into the intermembrane space. And then we need to talk about facilitated diffusion and how H+ ions move back in, which causes a change in shape of the ATP synthase, which results in the formation of ATP. And then we also mentioned oxygen is the final electron transporter, which causes the movement of the electron from carrier protein to carrier protein to eventually get to the oxygen. So those would be your 6 marks. And you would also get marks for other things, but I'm just saying what I would write for these 6 marks. So your 6 points. Okay. Sorry, we're rushing through these, but I don't want to hold you guys here any longer. And then this was quite another easy question, so we'll just skip over it. So it's just asking about the products of ethanol fermentation. I'll just ask you a quick question, actually. What is the product of glycolysis? The main product of glycolysis, even. Yeah, pyruvate. Just making sure you're still with me. So in this question, I thought it was quite interesting because the scientists were looking at the effect of an enzyme inhibitor, malonate, in the Krebs cycle. And so they decided to add malonate and the respiratory substrate pyruvate to a suspension of isolated mitochondria. And so the question was, why can't you use glucose instead of pyruvate for these as the respiratory substrate in this reaction with the isolated mitochondria? So can anyone think of a reason why we can't? So can glucose enter the mitochondria directly? Yeah. So that's our main reason why we can't use glucose as our respiratory substrate. So glucose is actually broken down, is actually converted. So that's converted to pyruvate via glycolysis, which occurs in the cytoplasm. So if we only have isolated mitochondria in this reaction, we only have isolated mitochondria here, this means there is no cytoplasm, and so glycolysis can't occur. And so we can't have glucose directly trying to get into mitochondria either. So that's our second point, glucose cannot enter the mitochondria. Yeah, so glucose cannot enter the mitochondria. So that would be our 2 marks. I hope that makes sense. And then I think next question might be our last question. Yeah, that's our last question. So for our first question, it is explain why converting pyruvate to lactate allows the continued production of ATP. So can anyone give me 2 points that answers those 2 questions, that question? So what do we get from oxidizing reducing pyruvate to lactate? What do we regenerate? What's the important coenzyme we regenerate? Yeah, so from converting pyruvate to NAD, we are regenerating NAD. Regenerating NAD. And then as this happens, so if you remember the reaction, the reduced NADH is going to NAD. This means the NAD that we've regenerated can re-enter glycolysis to generate ATP. So our 2 marks in this question would come from 1, the regeneration of NAD as the NADH is oxidized to NAD. And then the second mark is NAD can then enter glycolysis to generate our ATP. So our very last question is that some of the lactate is converted back to pyruvate when there is enough oxygen. Can anyone give me an advantage of doing this instead of just keeping the lactate in our body like it is? So we can look at it in 2 ways. So the one way would be regenerating the pyruvate is good for us because if we have enough oxygen now, the pyruvate then can do aerobic respiration, can then enter aerobic respiration. And that can be one way to look at the question. Another way to look at the question might be, why is it bad to keep the lactate in our body? So lactate buildup in our body is an acid, so lactate acid buildup, it decreases the pH and can denature our enzyme, which is also bad for us. So these are 2 different reasons why it's good that the lactate gets converted back to pyruvate, but it's just different ways of looking at it, and you'd get marks for either of these. So I hope we've covered everything that we said we will in this talk, and you're still awake at this point. So I hope you enjoyed and thank you for listening. Please fill in the feedback form at the end. And I'm sorry for overrunning. If you want to vote one last time, if you understood everything, if you put it as a ranking, so A is you understood everything and D is you didn't understand anything. Oh, that's great. Thank you, guys. I'm happy to hear you've understood everything today. Please fill in the feedback form as well. Thank you. Sorry for overrunning, by the way. Tamkin?